Hi, everyone. My name is Geneva Wang, and I'm a technical product manager for CyTOF reagents at Standard BioTools. I'm excited to walk you through how LyoMax CyTOF panels helps researchers standardize high-parameter assays at scale, particularly in large-scale longitudinal and multi-site studies. Let's start with what LyoMax delivers. So LyoMax CyTOF panels take your unique custom liquid panel and transforms it into a stable lyophilized assay. This enables standardized immune profiling across time points, cohorts, and sites. We offer a 50 marker capacity covering both surface and intracellular targets, customized based on your research needs. The panels can be manufactured at standardized lot sizes of 100 or more tubes, ensuring consistency and, most importantly, reproducibility. The single tube dry format means you can just add samples for a simple, streamlined workflow. Why do high parameter panels and measuring more matter? This is because the mechanisms and biomarkers driving therapeutic responses are increasingly complex, and we see this repeatedly in published studies. In a recent phase 1/2 multiple myeloma trial, the MaxPar Direct immune profiling assay, our off-the-shelf 30 marker lyophilized panel, was customized to evaluate the response following TIGIT LAG-3 blockade in order to understand their immunologic effects and safety. So using the MaxPar Direct immune profiling assay, changes in LAG-3 expressing T cells between partial responders and non-responders were revealed. This approach also enabled the characterization of unique cellular pathways upregulated in patients receiving anti-TIGIT. On the right, you can see in another study, intracellular staining with CyTOF technology ensured accuracy in detecting IL-10, along with 30+ immune markers to capture IL-10 positive regulatory B cells. And these cells were associated with high-grade immune-related adverse events. So to summarize, immune profiling has emerged as a critical tool to reveal predictive biomarkers. With high parameter panels comes operational complexity. In large-scale or multi-site studies, one of the biggest challenges is reproducibility due to lack of standardization. Many studies today face reagent variability between lots, inconsistent SOPs across operators, stability challenges, and limitations at remote or resource-restricted sites. So logistical complexity adds time and cost, but variability affects data quality, marker intensity, and resolution. And this is where LyoMax can transform workflows. So compared to liquid CyTOF panels, LyoMax panels require less manual work and deliver equivalent performance with valuable time savings. On the left, you can see all the reagent prep required with liquid cocktails. With LyoMax, simply add the sample. This reduces prep time by over 2 hours for a 30 marker panel. In this plot here on the right, you can see that we compared 3 independent operators, each pipetting 3 of the same liquid panel, and we compared this to the CVs of the LyoMax panel. The LyoMax panel resulted in lower CVs compared to the liquid panel across operators. So this means better reproducibility and less user-to-user variation, which is critical for multi-site studies. LyoMax transforms liquid antibody panels into a stable dry format, eliminating variability and manual preparation, and also ensures reproducibility. So LyoMax panels are built on the same foundations as our trusted MaxPar Direct immune profiling assay, which was launched in 2019 and used in hundreds of studies and clinical trials. With less than 10% CVs between sites and instruments, these panels are easily expanded and compatible with stabilizing reagents and barcoding methods. Just like our MaxPar Direct immune profiling assay, when appropriate clones are chosen, LyoMax panels are compatible with stabilizing reagents and can provide up to 72 hours of reliable results with stabilized whole blood. This compatibility ensures accurate immune profiling even with delayed sample processing or transport, simplifying logistics for remote or asynchronous sample collection. LyoMax panels work seamlessly with live cell and Palladium barcoding. So by barcoding, you can pool samples for staining, and this minimizes staining and acquisition variation and streamlines processing. In addition to enhanced consistency and reproducibility associated with the use of LyoMax panels, multiplexing further enhances batch-to-batch consistency and reproducibility. So now I'd like to highlight an exciting study coming out of the University of Sydney. And in this example of a collaboration with the Maguire Lab at the University of Sydney, researchers developed a workflow that enabled them to stain with a 38 surface marker, custom-tailored LyoMax panel at 3 different collection sites. They were able to freeze and store samples until time for shipment to a central processing center. The total upfront processing time was about 40 minutes. At the processing center, samples were then barcoded and stained with additional intracellular targets and acquired. This assay was chosen as it required minimal training and could be implemented at remote sites in Australia. Their conclusion, CyTOF is the only technology that allows easily implemented remote asynchronous sample collection and the largest immunophenotyping panel that yields reproducible results. CyTOF's wide coverage with panels routinely containing 40, 50, and even 60 markers allows researchers to capture unique biomarkers. With spectral fluorescence, one can miss critical differentiating markers. But with CyTOF, you can simultaneously analyze surface and functional markers, uncovering predictive responses and mechanisms that fluorescence-based flow cytometry may overlook. CyTOF technology captures the breadth and depth of immune coverage required for identifying unique biomarkers. So you might be wondering why CyTOF is able to capture differences compared to spectral, and the difference is due to signal quality. At the top of the slide, you can see the emission spectra for several fluorescent dyes. This illustrates that there is significant overlap between emission spectra of fluorescent tags. This overlap requires spectral unmixing or compensation, and these are affected by instrument configuration settings as well as the use of appropriate controls. These samples may also potentially require autofluorescent extraction, and all of the above make it difficult to accurately identify low-expressing targets. CyTOF technology uses metal tags, which means minimal overlap, and signals that are predictable and signals that have similar intensity. With metal-labeled antibodies, CyTOF technology allows for comprehensive profiling of many cell types and functions. Here's an example of what CyTOF enables. In this 61-parameter panel, it was able to capture activation, differentiation, exhaustion, immune checkpoints, and intracellular cytokines. Panels like this offer comprehensive systems-level insights from a single tube. So to summarize, with LyoMax panels, you get a single tube workflow, just add sample. A fully customized high-parameter panel with up to 50+ markers. You get standardized batches of 100+ tubes, ensuring batch consistency and reproducibility across sites and operators. And lastly, you get support from our team at every step. With LyoMax, researchers are supported end-to-end by our service lab team, and this includes panel design and liquid panel optimization to biological assay validation of the lyophilized product, which includes evaluation tubes for assessment before manufacturing of the final product. Our team has completed hundreds of service projects globally and are ready to support every step of the way. Thank you, everyone, for joining today's masterclass, and please don't hesitate to reach out if you have any questions. Thank you.